All right, let's get started. So thank you to the Alberta Mycological Society for hosting this lecture for the second time. And a special thanks to Zoe Henderson, who is helping out as a co-host tonight. And it's her birthday, so triple thanks to Zoe, and happy birthday. Tonight I'll be speaking on the mycelial material revolution, which is ultimately a shift in manufacturing where fungi are now being used to produce a host of new products with novel properties. Applications can be found currently in packaging, textiles, food, fashion, construction, but that's just the beginning. This is a time of innovation, and over the next couple of decades, I think we're going to see some startling new things. The talk will be broken into three parts. First, I'll go into the basic background science behind the revolution. In the second part, I'll talk about the cultural history and current state of the revolution, as well as the main players. And in the third part, I will present my proposed response to the revolution, which will take the form of a hybrid community lab with a little meandering here and there throughout. So bear with me. First, I'd like to dedicate the talk to Dr. Roy Crawford, a mad scientist of the chicken variety, but also a master teacher. The Ralston Purina Award is an international teaching award, and a master fermenter of the pickling variety. His major contribution to poultry genetics was the discovery of a mutated gene that causes epileptic seizures in chickens. As silly as this sounds, the discovery is still an important animal model in human epilepsy today. And how does one determine whether or not a chicken has epilepsy? From my understanding, it involves a dark bunker and a strobe light. So I currently live in Calgary, but before moving to Calgary in 2020, I worked at GNC Bioferm, which is a biotechnology company incorporated in Saskatchewan in 1985. We use fungi to develop feed enzymes for swine and poultry, and some rabbits, I think. Some fish too, mostly swine and poultry. So what is a feed enzyme? It's a specialized protein that helps break down starches, fibers, and even other proteins into simpler forms. This helps improve the nutritional uptake of the animal feed, as well as cleans their waste. It helps farmers get more bang for their buck. The fungi we grow produce these enzymes naturally, which we harvest and sell as a feed additive. If you look closely, this is me on the lawnmower. I find cutting grass meditative. I was actually the lab manager at GNC though. So here's a shot of one of our bugs, which is a mutated form of Aspergillus oryzae growing on a trade secret. Aspergillus oryzae is one of the earliest domesticated fungi used for koji and sake production in Japan. It's a close relative of Aspergillus flavus, which can be found almost anywhere. Our version of Aspergillus oryzae is designed to grow slow and overproduce digestive enzymes, making it especially susceptible to a vast array of competing organisms that look nearly identical but grow much faster. It's important to note that since fungi show Mendelian inheritance, going from flavus to oryzae plus is kind of like dogs for selective traits. It doesn't require like CRISPR technology or anything fancy. When I wasn't combating these contaminants, I ran experiments in an attempt to maximize the enzyme output of our molds. We ran tests on different types of nutritional sources. 1 second. Sorry, I saw something in the chat. It wasn't for me. We ran tests on different types of nutritional sources and growth environments, moisture, and temperature. Enzymes tend to come in bundles when they are produced biologically, but here are the three main ones we were interested in. So there's amylase, which is an enzyme that breaks down starch into simpler sugars. It's found in human saliva, and you can actually taste it at work when you're eating rice. If it's slightly sweet, that's the amylase doing its thing. Xylanase, which breaks down xylin, major structural component of the cell walls of most plants, and beta-glucanase, which breaks down beta-glucan, a common dietary fiber and main constituent of the cell walls of cereals, most cereals. The quick rule of thumb for enzymes, the suffix "ase" generally means you're dealing with an enzyme, and what comes before that is what the enzyme breaks down. So before working at GNC, I pursued a Master's of Science at the Institute of Computational Design and Construction in Stuttgart. This institute was founded by Professor Achim Menges in 2008. And through the institute, students were taught an approach to design that combines biological, material, and fabrication research to produce novel architectural forms, but more importantly, novel architectural processes. The three research pavilions seen on the right are the products of this approach to design. It's important to note that the forms that emerge are as dependent on the biological models that inspire them as they are on the materials used and tools used to mold materials. So on the bottom right is just as an example. The biological model of that pavilion was a subaquatic spider that weaves its nests in a bubble just under the surface of the water. Carbon fiber was the material, and a robotic arm was used to laminate the carbon fiber to an inflated bubble. The point being that all these three things like connected to produce that form. It's not just the biological model, and it's not just the carbon fiber, but an interaction between all three. So the ICD is the institute, and the ITEC is the actual program. So ITEC stands for Integrative Technologies and Architectural Design Research. It's a master's levels program offered by the ICD in partnership with the ITEC, which is run by Professor Jan Knippers. The program is broken into two parts, with the first year being dedicated to producing a research pavilion, like the ones described above. This was my year's pavilion. The biological model was a sea urchin, and thin plywood was used as the construction material, where with the fabrication method being robotic sewing. The key takeaway is it's teaching, they taught science and technology as like a method of craft, like hands-on. In the second half of the ITEC program, the students pursue their own research interests. I went with fungus. My research culminated in a paper entitled Modular Mycelia, Scaling Fungal Growth for Architectural Assembly, that I presented to the Regional eCAADe Symposium in Wales in 2017. The overarching hypothesis of this research was that fungal growth could be used for architectural application through the development of a multi-scalar assembly system. Initially, the spheres were designed for pouring. I wanted it to be able to pour it like concrete, but we tried to move away from formwork, which was, in retrospect, was kind of unfortunate because it also left behind the potential for designing a new kind of formwork made specifically for living systems, which could have been interesting and something I will probably still pursue down the road, patent pending. So again, the context of the main body of the talk are 1, the science, 2, the culture, and 3, the lab. So here's a video of Hyphae growing. The takeaway from this is that growing occurs at the apical regions of the mycelium, or the tips, and the bright point that you're seeing here is known as the Spitzenkorper, which is the organizing center for hyphal growth. That's where all the magic happens. So this is a simplified phylogenetic tree, a diagram that depicts the lines of evolutionary descent of different organisms. The fungi represent a heterogeneous group of eukaryotic microorganisms, and it's heterogeneous because it's so vast, it's very hard to find boundaries, taxonometric boundaries. Interesting factoid, fungi were once classified as plants, more specifically as a type of albino algae that had lost its chlorophyll through evolution. They are fundamentally different than algae, though. Fungi are mostly terrestrial, while algae are found in aquatic regions, although there are certain types of fungi that can be found in freshwater or even marine environments. Since fungi do not contain chlorophyll, they do not require light to grow, while algae do. So this is a simple Venn diagram illustrating the differences and similarities between plants and fungi. Where the plant cell walls are made of cellulose, the fungi cell walls are made of chitin. Another difference is that while plants have roots, the fungi have hyphae. Plants are autotrophic, which means that they produce their own nutrition. More specifically, plants are photoautotropic, meaning that they use photosynthesis to produce energy. Fungi are heterotrophic, using mostly organic carbon and nitrogen as a food source. This means that fungi rely on external sources of nutrition like animal or plant matter. This also means that they play the role of decomposer in nature. This will become important a little bit later on. How do you make material out of a decomposer? So this diagram shows the basic anatomy of a basidiomycete, also known as a club-forming fungus. It's important to note that the club refers to the shape of the spore and not the mushroom. Basidiomycetes are mostly terrestrial with wind-dispersed spores, but some grow in freshwater or marine habitats, as I said. Note that the septae that were missing in the previous video, those are the partitions like along the hyphae. Most of the Mycelium and I think even the Ascomycota have septa. So I'm not sure what we were quite looking at with that previous video. They didn't seem to have them. These septae have a central pore which maintains cytoplasmic continuity between adjacent segments. While there is movement of most organelles through the septal pore is permitted, the passage of nuclei is not. This is possibly a consequence of their larger size of the Basidiomycota. In the Ascomycota, which was previously shown, that's the green molds, the non-mushroom fungus, their nuclei are smaller, so they can actually pass through the pores. So this is these diagrams show the basic anatomical features of the fungi's hyphae. Hyphae are the filamentous cells of fungi. They are composed of microtubules, nuclei, vesicles, and chitosomes. Growth, communication, and digestion occur at the tips of the hyphae, guided by the Spitzenkoper, which we saw earlier. Mycelium tend to grow in radial patterns because the enzyme and chitin production occur at the apex, the tips. So the chitosomes produce the chitin and the vesicles produce the enzymes. So those like animations, I had to do some quick editing right before the presentation started. So the different types of Basidiomycetes are often distinguished from one another by the shape or color of their fruiting body or mushroom, though the material characteristics of the mushroom's mycelium can vary drastically across species as well. So here we have a close-up shot of the mycelium mass growing on wood. It's just oyster mushrooms, Pleurotus ostreatus. And here we have an even closer shot of the fungal hyphae growing through and creating a mechanical connection with the substrate. It's important to note that these microphotos were taken on a slide microscope, not like the previous digital microscope, which means that the mycelium network is misshapen. It was kind of torn apart and placed under a slide, so it's not quite accurate. But you can still see the hyphae growing into the wood, which I thought was really interesting. So this is a conceptual graph showing the role of fungi as a decomposer is an important consideration. It means that if left alive, fungal-based material will eventually decompose. Another important point of consideration is that before a fungus decomposes a substrate, it first grows through and binds it as a source of nutrition. It's this capacity of growing around and through a substrate that make fungi-based materials viable. Fungi-based material will hit a peak point of structural performance during colonization, after which the fungus will weaken the material through its enzymatic digestion output. So this is a close-up of a mycelial mass growing on a plastic mesh. I also added a nutrient-rich agar to make sure, because the plastic itself wouldn't provide enough nutrition, I don't think, for it to grow properly. And here's an even closer shot of the fungal hyphae growing on and eating away at the plastic mesh using its digestive enzymes. This is important because apart from making materials, fungi is also really good at decomposing plastic and even remediating like oil ruined land. So this is Neri Oxman, who's a designer and architect and professor at MIT's Media Lab. She couches her research in the field of material ecology, often using biological models as a source of inspiration in actual materials. This is one of a number of research projects using chitin-based composites as a printing material. This is an example of one of them. It's a big wing-looking thing. So, chitin is a fibrous polysaccharide, second most abundant polysaccharide found in nature next to cellulose. Chitin is also found in the exoskeletons of crustaceans and insects and in the cell walls of protozoa. In nature, chitin is often combined with other minerals or polymers to produce composite materials. An example is the shell of an arthropod, which is a composite of chitin and calcium carbonate. Fungi mix chitin with glucan to form rigid cell walls. People don't produce chitin. The closest thing we have is keratin, found in our hair or nails, but it's actually a fibrous protein and not a polysaccharide. Okay. So as I said, Neri Oxman is 3D printing with chitin, but this is an oversimplification. You can't really 3D print effectively with chitin. Shrimp shells aren't particularly soluble, and it'd be difficult to produce a printable material with chitin alone. So Oxman is actually printing with chitosan composites, which is a soluble derivative of chitin, seen here. To extract chitin from shrimp or crab shells, they are treated with acid to dissolve the calcium carbonates in the shell, and then treated again with an alkaline substance to make the remaining proteins soluble as well. Decolorization step may also be added to remove leftover pigments and produce a colorless product. So here we see some chitosan gradients. Much like the way crabs or grasshoppers make their shells and wings, these chitosan gradients are actually biocomposite hydrogels with cellulose, pectin, and I think potentially calcium carbonate mixed back in. These hydrogels are then distributed using a multi-axis KUKA arm on a linear thread with a 6-headed extruder that can rotate. Important to note, while chitosan is easier to process than chitin, it's less stable, not only due to increased water solubility, but also pH sensitivity. The revolution will be pasteurized. Why? You can print with chitin, straight chitin, not chitosan, without all these extra steps, simply by controlling fungal growth patterns. So, the culture. What we're seeing here is an advertisement from Ecobative, which is they're basically the Apple of the mycelial revolution. I think this is specifically a division called Forager, which is a breakoff focused on not on biocomposites, which would have a substrate, but with straight mycelia. And it's for foam and a fashion things. Foam for fashion and also producing. We'll get to it later. There are many possible products, as there are as many possible products as there are different kinds of mushrooms, which we're kind of seeing at the end of the video. It's kind of interesting on that right, they end with Amanita muscaria. That's the Mario or Alice in Wonderland mushroom. So this image is rice with Aspergillus oryzae growing on it. Same as, not the mutated version, but that's what we were seeing. That's the green petri dish from earlier on in the presentation. The Japanese used it to saccharify complex carbohydrates found in rice to make koji and sake. So this is an illustration of a traditional sake fermentation setup. And I look at this and like, how did this not result in massive contamination? This, and more importantly, this is an early example of synthetic biology, where the natural growth processes of fungi are co-opted to produce food and alcohol, a precursor to the current revolution. So The discovery of penicillin in 1928 by Alexander Fleming kicked off the medicinal fungal revolution, where fungi were used to produce medicine. Folk medicine already had an in on the antibacterial properties of fungus. The Chinese discovered that moldy soybean curds could be used to treat foot boils circa 500 BCE. The fungicide converted the simple sugars from the curds into a rudimentary antibiotic. However, Fleming's discovery started a period of interest in mutation induction and analysis of mutants for biochemical traits, much like the enzyme producing molds of GNC. From food to agriculture to medicine to materials. So this is a research project conducted by Eric Clarenbeek entitled Mycelium Chair in 2013. In it, he's 3D printing with live mycelia as opposed to chitin hydrogels, which then are allowed to grow, and as they grow, they strengthen and structuralize the chair. I think the bursts of fruiting bodies were actually an accident. They claim that it's an ornamental part of the chair, but it acts as a proof of concept. Mycelia is indeed alive. And to the left are just research samples to develop the chair. It's kind of interesting that artists and designers are playing a major role in this revolution, where the previous revolution was mainly relegated to the laboratory scientists. An example of this is Philip Ross, who's an artist and inventor. He co-founded Microworks in 2013, which is one of the major players in the revolution. It's a company that produces biomaterials out of mycelium, most notably leather. He currently holds the position of Chief Technology Officer at the company, but has been producing fungus-based works since the 90s. That's him holding a block of reishi. So this is an example of one of Ross's fungal sculptures. I think it's from the late 90s or early 2000s. A lot of these projects are long-standing, so it's hard to find specific names or specific dates. But it's an example where fungus-based material was used as an artistic medium to create temporary sculptural forms. And this is a project, our Ross project from 2013. Full title, Mycotextural Polyominoes. So in the first step of this project, a mix of reishi, mycelium, and sawdust is packed into a mold. And then after a few days, a block is formed. As the project title suggests, these blocks take the form of a polyomino, which is a planar polyform made out of squares. They're often used for problems or puzzles that involve a tiling plane. Tetris is a classic example. Dominoes is another one. The project is also a demonstration of how the fungi's anastomosis, or hypobonding can be used as an assembly technique. After the mycelial blocks are formed, they are put into close physical contact with each other, and eventually the blocks fuse and a continuous object is formed. An interesting application of a natural growth process. So this is a clip from Microworks demonstrating their material platform, Fine Mycelium, which is a patented process that engineers mycelium cells as they grow to create interlocking cellular structures. This is unlike other versions of fungus-based leather, which use compressed mycelium. The process can be tuned to produce a range of material properties. So in 2017, Microworks partnered with Hermes to create Sylvania, a custom version of Microworks Fine Mycelium. Hermes and Microworks spent 3 years experimenting with this process, which could be considered an exemplar of a new generation of fungus materials. Sylvania will be manufactured in California and then shipped to France, where Hermes will incorporate it into a range of products. So it's important to note, Sylvania is what the actual leather is called, not what the purse is called. Or bag, sorry. This collaboration heralds a broader cultural acceptance of fungus-based materials as superior to and not simply more sustainable alternative to more conventional materials. This is a growth study I conducted using simple oyster mushrooms. Not so much tuning, because I kind of did it by accident, but the principles are the same. Tropic manipulation. So tropisms are usually associated with plants, think sunflowers turning towards the sun. Fungi also display this form of responsive motility. Some of the tropisms that affect fungi growth patterns are aerotropism, which is a growth response to oxygen, autotropism, which is a tendency to grow in a straight line in the absence of other tropisms, chemotropism, which is a response to chemical stimulus, galvanotropism, which is a response to electrical stimulus, gravitotropism or geotropism, which is a response to gravity, and thigmotropism, which is the movement or orientation of hyphae in relation to the topography, shape, or physical properties of the underlying substrate. So the point being, there are a lot of options in terms of tuning your material properties or controlling the growth processes of fungi. So Ecobative is a fungus-based material company located in New York. It was founded by Eben Bayer and Gavin McIntyre, seen here. They actually founded the company as students at the Rensselaer Institute, and the initial idea was to, I'll get that later. So Ecobative currently provides a range of sustainable alternatives to plastics and polystyrene foams for packaging, building materials and other applications through mycological technology. Okay. So the flagship product was a custom molded secondary packaging. Secondary because the wine bottle is still in a bottle. Sorry, the wine is still in a bottle. They're not certified for direct contact with foodstuffs yet. Fungus-based packaging is actually a biocomposite that uses an agricultural byproduct, corn husks, as a substrate. So the initial idea that they had was to compete with Dow Chemicals by replacing styrofoam with a product that can be easily composted. So after you receive your packaging, the idea was you could just break it up and throw it into your backyard, and it would break down and actually potentially improve the quality of the soil instead of being stuck forever in some landfill. Ecobative now produces a range of materials used in packaging, building materials, rigid boards and insulation, food products, fashion products, foams and leathers, and meat alternatives. Note that Ecobative mostly develops the materials. The actual products come about through collaborations with other companies, just like Hermes and Microworks. So I think it should be mycological matrix composites. So mycological matrix composites is the title of a paper that was presented at a conference for the American Society of Composites in 2013 and 2014. The book was published in 2014, but the conference was in 2013, I think. It concluded that fungal-based materials have a host of novel mechanical properties. They are lightweight, which increases ease of transportation and ease of assembly. They have lower dead loads. They are durable and can absorb impact loads and fail non-catastrophically, unlike masonry or concrete. They are flame resistant and self-extinguishing. And fungus-based products have been shown to have 2 to 8 times less conductivity than concrete and 10 to 40 times less thermal diffusivity than concrete, meaning they're naturally insulating. Oops. So the paper concluded there was a range of possible applications for fungus-based materials, from furniture to even aerospace composites, which I thought was kind of interesting. Point being, what we are currently seeing in terms of these emerging fungus-based products is only scratching the surface of fungi's potential. In 2019, Ecobative announced the launch of the Mycelium Foundry, an advanced research facility dedicated to mycelium materials. Through the use of custom-built solid-state fermentation instruments, and it's actually kind of interesting, solid-state fermentation is also how we produce our enzymes. And advanced tools for data analysis. So the Mycelium Foundry will allow Ecobative to accelerate the progress of these fungus-based products and materials significantly. Eben Bayer of Ecobative estimates that the Foundry will accelerate the rate of fungal-based technology by 10-fold. It's important to note that though the facility is new, the term like mycelium foundry, this is kind of confused me because so the facility is being developed like in 2019, but they've been referring to the mycelium foundry like in the company, like on the website since like 2010 or 12. And so they also use the term the mycelium foundry to refer to the tool set, the innovative tool set that they've developed as a company and not just the building itself. Section 3, the lab. So this is a quick fly through of the Salk Institute for Biological Sciences in San Diego, California. It's a design inspiration. I actually visited the Salk Institute as a student, architecture student in Calgary. This is before Germany. We had a tour and it was inspiring, very inspiring. The initial idea for the institute came from Jonas Salk, who developed the polio vaccine in 1952. In 1957, Salk conceived a place where scientists could explore the basic principles of biology through intense collaboration. To achieve this, Salk hired Louis Kahn to design an open and unobstructed laboratory space able to adapt to the ever-changing needs of scientific innovation. Kahn achieved this by putting all the mechanical systems, electrical lines, piping, ventilation ducts in the floors between the laboratories, which you can see here. So this would be the mechanical floor, and this would be the laboratory floor, and then mechanical laboratory. In this design, collaboration, innovation are king and queen. And I think it's kind of interesting, the Vierendeel truss, which is this right here, often used for bridges, you'll see them in bridges, are what made these open spaces possible. So there's also no columns in the spaces below. So it's not just the mechanical systems. So another inspirational model for Calgary's future lab is Boss Lab, which is located in Boston, Massachusetts, and incorporated in 2016 as a nonprofit. A lot of their equipment was donated by local companies, and it's a prominent example of a do-it-yourself biology lab. So anyone can become a member, and the members range from entrepreneurs performing early-stage experiments to scientists doing PhD-level experiments to hobbyists with a passion for biohacking. So Boss Lab is on the vanguard of the open science movement, a movement dedicated to the dissemination of scientific research to all levels of society and education. Boss Lab is not alone, as these community labs are popping up all over the globe. There are already a few operating in Canada. These laboratories are mostly found in 3 provinces, Ontario, British Columbia, and Quebec. I think there's one in Lethbridge too, Amino Labs. None in Calgary though, or Edmonton. Another important note is the Public Health Agency of Canada hosted the first Canadian Do-It-Yourself Bio Summit on March 16th, 2016. This summit brought together the federal government with academics and do-it-yourself biologists to discuss safety concerns associated with the movement, but also to discuss how the movement is affecting technological innovation in Canada. So finally, my response to all these cultural trends. I formed a company under the guidance of MMP's entrepreneurial training program. This would have been in November of last year, named LeShroomery Inc. It's a fungus-based company with 2 wings. One wing is a biotech incubator focusing on developing consumer-oriented solutions for amateur mycologists and mushroom enthusiasts. The other wing of the company is a fungus-oriented educator dedicated to diffusing mycological knowledge and laboratory techniques. Both wings of the company will be housed by a hybrid community lab located in downtown Calgary. And through both wings, we hope to engage this revolution. First through education, and then through design and implementation. So, from an educational perspective, the lab that I'm proposing will approach mycological understanding from 3 different perspectives, with the hope of gaining a deeper understanding of fungus than a singular approach could offer. These perspectives are craft, forage, and laboratory knowledge. So from the laboratory's perspective, I don't think I need to go too far into it. It's kind of a given, isn't it? That's how we learn about science in the lab. But another important aspect is the forage. So these are specimens that were collected by the Alberta Mycological Society during their big annual foray, which was held in September last year, 2021, near Lac La Piche College. The event uses a citizen science approach to collect, identify, and record exciting finds. From an educational perspective, foraging gives the student a first-hand understanding of ecology from the mushroom's perspective. The student develops an intuitive understanding of the mushroom's natural habitat, where they are found, how they grow, and how they function in the wild. From a biotech development perspective, as Ecobative's Forager initiative has already demonstrated, in which you kind of saw in that advertisement, they actually go out into the woods and look for new types of mushrooms and test their material properties to see what they can take back and use. I was in a webinar with Gavin McIntyre 2 days ago, and he claimed that they had over 300 specimens that they were testing for different types of materials and developing new products with them. All right. So the second perspective, or third perspective, is craft. So this is a fungus-based project entitled From the Ground Up by Jordan Livermore, a student at the University of Calgary's architecture program. Blue oyster mushrooms and hemp twine serve as the primary materials studied in this research to find ways in which mycelium structures may be reinforced, formed, and grown in place. It's kind of interesting of mixing the hemp twine with the other substrate, like the ropes, because generally as a biocomposite, mycelium materials are very strong in compression, but very weak in tension. So adding hemp rope to it is a smart design decision. So various methods of mycelium growing were tested with various weaving patterns, enclosures, and formal logics. The final prototypes test the production of 2 reinforced column concepts and 1 reinforced wall concept. From an educational perspective, working through craft gives the students an understanding of mycology that cannot be found in the laboratory or in the woods. It's kind of hard to summarize in a slide, but the work involved, The work involved in harnessing a living organism for functional application will give the student a different kind of understanding, which I believe will reinforce the 2 other perspectives. Just curious, don't answer, maybe after. Has anyone read Richard Sennett's The Craftsman? It kind of deals with this, not necessarily with living organisms, but just craft in general, and it's a really interesting read. Through craft, a person will form a bond with the tools, materials you're working with, and that goes doubly if it's alive. So, in conclusion, a budding ecology of organizations. So right now, I don't have a lab quite yet, but I'm forming relationships with Fuse 33, which is a makerspace in Calgary, a large makerspace. And the hope is, as the company develops, we'll use their equipment for prototyping. So right now it's all shared memberships, and also through the AMS go on forays and potentially find new mushrooms to experiment with. And then also in a couple, 2-3 weeks, I'll be opening a restaurant with some friends of mine, which is located in a community gallery. But the idea that'll be like the first target market. We're gonna go after the chefs with a fruiting pot I've been designing.